Hello friends, welcome to my channel Easy Life Science. Here I am talking about a practical nitrate reduction test and urea hydrolysis test. So please subscribe my channel, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. First, nitrate reduction test. The nitrate reduction test is a biochemical test useful for identification of unknown microorganism. The aim of this test to determine the ability of some microorganisms to reduce nitrate NO3 to nitrite NO2 or beyond the nitrite stage. Principles of this test are: certain microorganisms like chemolithoautotropic bacteria and many chemoorganoheterotrophic bacteria can use nitrate as a terminal electron acceptor during anaerobic respiration. Anaerobic respiration, here a final electron acceptor is rather than molecule oxygen. It may be a nitrate, carbon dioxide, or a SO2. Here, in this case, the nitrate is a terminal electron acceptor during anaerobic respiration. In this process, nitrate is reduced to a nitrite by the action of enzyme nitrate reductase. Further, the nitrite is reduced either to ammonia or molecular nitrogen. The medium used for this test is a nitrate broth which contains 0.5% of potassium nitrate, that is KNO3. The reaction for this test is the nitrite in presence of hydrogen and electron by action of nitrate reductase enzyme, it gets converted into a nitrite, that is a NO2, and further this nitrite get converted into a ammonia or a molecular nitrogen, and this formed nitrite or a NO2 can be detected by using the two reagents, that is sulfonylic acid and alpha naphthalamine. Both these reagents are colorless, and in presence of nitrites they forms a compound sulfobenzene azo alpha naphthalamine, and this is a red colored compound. Anaerobic metabolism requires an electron acceptor other than atmospheric oxygen. Many gram-negative bacteria use a nitrate as a final electron acceptor at time of anaerobic respiration. The nitrate reduction test is the test that determines the production of an enzyme, that is a nitrate reductase, which results in a reduction of nitrate, that is a NO3. Bacterial species may be differentiated on the basis of their ability to reduce nitrate to a nitrite or a nitrogenous gases. In this test, the nitrite is formed from a nitrate, and the nitrite in presence of a sulfonylic acid forms a complex, that is a nitrite sulfonylic acid, and when nitrite sulfonylic acid reacts with alpha naphthalamine, it gives a red colored precipitate, that is a prontosil. It is a water soluble azo dye. To perform this experiment or to perform this test, a heavy inoculum of a test organism is inoculated in a broth containing a nitrate, for example peptone nitrate broth. The organism capable of producing nitrate reductase enzyme reduce the nitrate present in a broth to nitrite, which may further reduce to a nitric oxide, nitrous oxide, or a nitrogen. The reaction for this test, the nitrate present in a medium in form of KNO3 by action of a enzyme nitrate reductase get converted into a nitrite, that is a NO2. If in a medium there is a formation of nitrite, it can be detected by using the two reagents, that is a sulfanylic acid and alpha naphthalamine. In presence of sulfanylic acid or a reagent A, the nitrite choose a colorless compound, that is a nitrate sulfanylic acid. When we add alpha naphthalamine, that is a reagent B, in the medium, there is a formation of a red precipitate of a compound prontosil. Formation of prontosil or a red colored compound show that there is a formation of a nitrite and show that a test is positive. To perform this experiment, the requirements are a peptonitrate broth composition. It contains peptone, yeast extract or a meat extract, potassium nitrate, have to address a pH 7.0. The reagents like sulfanylic acid and alpha naphthalamine, and a 24 hours old culture of test organism. Procedure: at aseptic condition, by using sterile wire loop, inoculate the nitrate broth tubes with a given bacterial suspension. After inoculation, incubate the tubes at a temperature 37 degrees Celsius for 24 hours. After incubation, add 6 to 8 drops of nitrate reagent A, that is a sulfanylic acid, and add 6 to 8 drops of reagent B, that is alpha naphthalamine. If there is a formation of a red color after addition of reagent A and B in a broth tube, it says that test is positive. If no such color, says that test is negative. Here in this image, the left tubes is a positive, whereas a right tube is a negative. Uses: all the members of a family Enterobacteriaceae reduce nitrate, but some members further metabolize nitrite to other compounds. It is thus usual to differentiate a member of family Enterobacteriaceae that produce an enzyme nitrate reductase from gram negative bacteria that do not produce the enzyme nitrate reductase. The reduction of nitrate may be coupled to anaerobic respiration in some species. This test is used in a differentiation of a mycobacterium. It is also used for identification of some species of Neisseria and separating them from Moraxella and Kingella. This test is a critical test in differentiating between Neisseria gonorrhoeae and Klebsiella denitrificans. This test is also useful for identification of a species of a Corynebacterium. The next test, that is a urease test. It is also called as a urea hydrolysis test. It is an important biochemical test used for identification of the unknown organism, also for a diagnostic purpose to diagnose the urinary tract infection. Aim of this test to determine the ability of a microorganism to degrade urea by enzyme urease. Principles: the urea is a waste material, waste after a digestion of a protein nutrients. It is a diamide carbonic acid, often referred as a carbamide. Hydrolysis of urea is catalyzed by a specific enzyme, that is a urease. It yields 2 moles of ammonia. The enzyme urease attacks the nitrogen and carbon bonds in urea and forms ammonia. The presence of urease enzyme is detected by a formation of ammonia in a broth. So, in a medium there is an addition of a pH indicator, that is a phenol red. If ammonia is there, it says urease enzyme was there. The splitting of urea creates the alkaline condition which turns a phenol red to deep pink in color. This test is mainly useful for identification of a protein species from other genus of lactose non-fermenting enteric organisms. In this reaction or in this test, the urea it is hydrolyzed by an enzyme urease to a carbon dioxide and a ammonia. Interpretation: after inoculation and incubation, if the medium shows a deep pink color, then test is positive. Principle of this test: urea is a product of decarboxylation of amino acids. Hydrolysis of urea produces ammonia and CO2. The formation of ammonia alkalizes the medium and the pH shift is detected by a color change of a phenol red from light orange to a magenta pink. The rapid urease organism turns the entire medium pink within 24 hours. To perform this experiment, the scientist Christensen formulated a medium, that is a Christensen's urea agar, in 1946. The medium, that is a Christensen's urea agar, used for a differentiation of enteric bacilli. The urea test is used to determine the ability of organism to split urea through a production of enzyme urease. The composition of Christensen's urea agar: urea, sodium chloride, monophosphate, monopotassium phosphate, peptone, dextrose, pH indicated as phenol red, and agar. Uses of urease test: this test is used to differentiate organisms based on their ability to hydrolyze urea with the enzyme urease. This can be used as a part of identification of several genera and a species of Enterobacteriaceae, including Proteus, Klebsiella, and some non-Enterobacteriaceae like Yersinia, Citrobacter, and Corynebacterium species. This test is also useful for identification of a species from Cryptococcus, Brucella, Helicobacter. For this test, requirements like urea agar slant and a 24 hours old culture of the test organism. Procedure: at a aseptic condition by using sterile wire roof, inoculate the suspension of test organism on a urea agar slant. After inoculation, the slants are kept for incubation at a 37°C temperature for 24 hours. After incubation, observe for a pink color development in a tube. It says that test is positive. If the test is positive, then we can see for a deep pink color as shown in a figure. Development of intense magenta to a bright pink color in 15 minutes to 24 hours. Example of urease positive organisms like Proteus, Cryptococcus, Corynebacter, Helicobacter, Yersinia, and Brucella. No change in color says that test is negative. For example, Escherichia, Shigella, and Salmonella. Thank you friends, thank you for watching me. If you like my video, please subscribe my channel, share my video to your friends, and don't forget to press the bell icon for further notifications. Thank you once again.